I'm really deeply grateful and honored to receive this wonderful prize. Thank you to the Vassar Center who have done such incredible work to support basic research and also the clinical care of patients with this mutation. I want to also, of course, thank the Gray family for the support. Actually, until last week, I didn't know if I could receive this prize coming from NIH and the bureaucracy at NIH. But it took 1 year with many emails, Zoom meetings, repeated questions and answers. And so I want to thank Beth and Susan for their patience. But finally, I can accept it officially. Let's see, I'll start the first slide. And before I start with my basic research to describe the research that we've done, I just wanted to say that even before I started the research, I knew of the importance of this area. In 1973, my mom was diagnosed with breast cancer at the age of 45. She was treated, and actually she lived till 91 years old. My sister, in 1994, was diagnosed with breast cancer as well. And 1994 was a very special year. My sister Sonia is not a scientist, but somehow or other, she knew about Mary Claire King's discovery of BRCA1, and she found out that she could get genetic counseling and that she could get tested. And indeed, she got tested and found out that she was a carrier for BRCA1. So I'm really proud of her. She's one of the only non-scientists in the family, but she did this. She did this research herself. I'm also really honored because I'm following, as you mentioned, there were 10 recipients of the Global Prize. And our work really was influenced by these leaders in the field. I mean, of course, Mary Claire King enabled my sister to get tested for this mutation. 1 year later, Alan Ashworth and Stratton discovered the BRCA2 gene. And then work from David Livingston, Ashok Venkataraman, and Maria Jason figured out how BRCA1 functions, and that it functions in double-strand break repair and homologous recombination. Alan and Thomas Halliday discovered a synthetic lethal relationship between BRCA1 deficiency and PARP inhibitors, which are obviously used in the clinic and been very successful. And David Livingston discovered PALB2, that PALB2 was also a tumor suppressor. And in addition, together with Roger Greenberg, they discovered chromatin complexes associated with the BRCA genes, which Roger further enabled him to figure out ways to potentiate PARP inhibitor treatment with these chromatin remodeling compounds that would inhibit the chromatin remodeling complex. So, but before BRCA was discovered, before PARP inhibitors were found to be to kill BRCA-deficient cancers, there were compounds well known in the clinic that already exploited synthetic lethality, that they were very, very potent and could lead to curing of some diseases. So this is cisplatin. It was used, I think, in the clinic in 1978. And it was found, by chance, to be very, very effective in certain types of cancers, particularly ovarian cancer and this type of ovarian, high-grade epithelial ovarian cancer. And then looking back, we realized why cisplatin is so effective. And the reason it's so effective is because at least 50% of these tumors have a defect in this pathway that I'm going to be talking about, this homologous recombination pathway, or what Agata mentioned, the Fanconi anemia pathway. And these proteins are important for repairing DNA damage induced by cisplatin. So in their absence, cisplatin is very, very toxic and will kill the tumor cells. But, and the other thing to really highlight that cisplatin and PARP inhibitors are so toxic to homologous recombination deficient cells is the fact that when these cells eventually become resistant to these compounds, they actually reactivate the pathway that was initially inactivated. So initially, they're homologous recombination deficient, and the major mechanism for resistance to cisplatin or PARP inhibitor is that the cells find a way to reactivate homologous recombination, and the cells become then resistant. So this is a tumor cell intrinsic, these are tumor cell intrinsic responses to chemotherapies, PARP inhibitor and platinum. But recently, there's a lot of interest in tumor extrinsic therapies, which utilize anti-tumor immune responses. And one way that Serena mentioned is the utilization of anti-PD-1 inhibitors that trigger the immune response to the tumor. And normally, the PD-1 interacts with its ligand, PD-L1, on the tumor cell, and this inhibits the immune response. And the use of anti-PD-1 antibodies enables the activation of the immune response to trigger immune attack. And just like homologous recombination deficient cells that become resistant to chemotherapy, there are a set of patients that respond to anti-PD-1 therapy and a fraction of patients that do not respond to immunotherapy. This is an example for melanoma. And there's a lot of interest, a great deal of interest to be able to predict, a biomarker to predict which patients would respond, which patients would not respond. And one major mechanism for response is the production of antigens on the tumor cell. So neoantigens that would be seen by the immune system. And one way that this can happen is by deficiency in DNA repair. So mismatch repair deficiency leads to a lot of mutations. This would produce a lot of neoantigens, and these are seen by the immune system. Absence of homologous recombination, in theory, could also be more prone to trigger an immune response. But in this paper, in melanoma, they figured out another way, independent of neoantigen burden, that would distinguish between patients that would respond or non-respond. And this was a transcriptional response. They could divide patients into non-responders and responders, and they found that certain transcriptionally overexpressed active pathways, such as wound healing, TGF-beta signaling, hypoxia, angiogenesis, and EMT, which we've heard from Agata, predict the non-responders, and then the decrease in expression of these pathways are more predictive of responders. So in that light, my talk is going to be divided into two parts. The first part is our figuring out how to tune the DNA response, DNA damage response in BRCA-deficient cells that are homologous recombination deficient, going from low homologous recombination repair to high homologous recombination. And the second part is kind of like the opposite, and it has some unpublished data that I would greatly appreciate your feedback on targeting homologous recombination proficient cells by tuning down homologous recombination. And again, I mentioned that homologous recombination reactivation is a major mechanism of chemotherapy resistance. So this is, I think, an important problem is how you treat the majority of the breast cancers are not homologous recombination deficient. So can you manipulate this pathway to better treat the homologous recombination proficient tumors? All right, so the key observation that started our work was work from Ralph Scully and David Livingston. It was a very simple experiment. This is one of the experiments they did, is they irradiate cells, and they look at the localization of BRCA1. And you can see here in red that BRCA1 forms these little dots. These dots are sites of DNA damage. And so BRCA1 is recruited to sites of DNA damage where it effectuates DNA repair. So this is the first indication that this is a DNA repair protein. And because of that, there was a lot of interest to make mouse models to see the tumor suppressor functions of BRCA1 and BRCA2. So could they make mouse models and generate tumors and try to understand the tumorigenesis, the mechanism of tumorigenesis? So this at first was a little disappointing or unclear what was going on, because if several groups knocked out BRCA1 and BRCA2 in mice, and what they got is embryonic lethality. So this pathway, homologous recombination, is essential for embryonic development. And meanwhile, Maria Jason used reporter assays to figure out how these proteins function in homologous recombination, how these proteins function, and concluded that they were part of the homologous recombination pathway. And this paper also showed a link with homologous recombination in that the BRCA-deficient embryos were hypersensitive to DNA damage. They can't repair the DNA damage. And at that time, a commentary came out in Nature from Kinsler and Vogelstein suggesting that loss of BRCA1 or BRCA2 were not sufficient to promote cancer. By themselves, they could not promote cancer. You needed to lose another pathway, a so-called gatekeeper pathway, for example, controlled by P53. So loss of DNA repair was, it led to genome instability, but you needed another deficiency in a checkpoint like P53 to actually get the tumors. And so so there were, two known major double-strand break repair pathways, one called non-homologous end joining and the other one homologous recombination. And the question in the field at that time, are these pathways really gatekeeper pathways that need loss of P53 to promote tumorigenesis? So briefly, non-homologous end joining is a very simple pathway because you have the DNA ends. They're recognized by the Ku protein, and then the ends are ligated together. Homologous recombination, unfortunately, is more complicated. And the ends are first processed to generate three overhangs, single-stranded breaks, single-stranded ends here, 5' to 3' resection. And this is a pathway. This step is controlled by BRCA1, this first resection step. And the reason that you need this single-stranded DNA is that you need to load the recombinase, RAD51, the key recombinase that will allow for strand invasion and repair off the undamaged template chromatin. So that's why you need the single-stranded DNA. And so Yas decided, since the knockout mice were embryonic lethal, couldn't do very much with them, but so he made conditional knockouts for BRCA2, and he knocked out BRCA2 in mammary cells. And he found that while loss of BRCA2 by itself did not lead to mammary tumorigenesis, actually I believe these tumors ended up not being BRCA2 deficient. So there are even, Old age. Old age. This is old age, but this is dramatically accelerated, real mammary tumorigenesis accelerated by loss of P53, according to exactly what Kinsler and Vogelstein proposed. And at that time, we were looking at the other pathway, the non-homologous, this more simple pathway. And so we knocked out this gene Q80, part of the complex that recognizes DNA ends. And we found that loss of Q80 also does not by itself promote tumorigenesis. But if you combine it with p53 deficiency, the mice get rapidly, they all get tumors, and all of them have chromosomal translocations. And you see that we were very influenced by that paper about gatekeepers and caretakers. At that time also, the question was raised, okay, what is the interplay between these two pathways? You have a double-strand break. How does the cell choose to repair it by homologous recombination or non-homologous end joining? One proposal is that they're just completely independent. For example, in the G1 phase of the cell cycle, maybe the breaks are repaired by non-homologous end joining. There's no sister chromatid available. And maybe in the SG2 phase of the cell cycle, the breaks now are repaired by homologous recombination. So to really study the interplay, the collaboration or competition between the pathways, there was work initiated by Maria Jason, and we're also on this paper, in which she crossed mice deficient in non-homologous end joining and homologous recombination. And what she found, and I don't think it would be very surprising, it wasn't very surprising to us, is that if you knock out homologous recombination component and a non-homologous end joining protein, the double mutant mice are much worse off. They were born at lower frequency than expected. There was a lot more DNA damage. And this increased damage implied somehow cooperation between these two pathways. So basically, it's bad to lose non-homologous end joining, bad to lose homologous recombination, and the combined deficiency goes from bad to even worse. This made a lot of sense until we and others started studying this other protein in the non-homologous end joining pathway, 53BP1. We had known that this protein repairs DNA breaks during immunoglobulin class switch recombination in G1 phase of the cell cycle. And when we did the simple Ralph Scully, David Livingston experiment, we and others, we found that the BRCA1 protein localized at different sites from 53BP1. They seemed to be mutually exclusive, as if some breaks are repaired by 53BP1 non-homologous end joining, some breaks repaired by homologous recombination. Maybe related to transcription, as Li Zhao mentioned, maybe transcriptionally active regions are repaired by BRCA1 and not transcriptionally active regions by 53BP1. However, Liu Cao in Torin Finkel's lab at NIH started crossing BRCA-deficient mice, this delta 11 allele, with various non-homologous end joining components. And as you would predict from the paper with Maria Jason, from bad you get worse. And that's what happened when he combined BRCA1 with all different non-homologous end joining mutants. They were worse off, more sensitive to DNA damage, et cetera, until by accident, really, and we can do these type of experiments at NIH. We got a lot of mouse facility, a lot of mouse space. He crossed BRCA1 with 53BP1. And amazingly, the loss of this other DNA repair protein rescued the lethality of the BRCA1 delta 11 allele. You have to really see. You see the mice at 1 month old. They look pretty normal. They were normal in all respects. And even at 7 months, they're alive. And these double knockout mice do not get tumors, unlike the BRCA1 p53 double E deficient mice. And so we proposed. The observations were that the viability is restored. BRCA1 deficiency is lethal. The loss of 5301 rescues the lethality. And homologous recombination is also largely restored. If we look at this marker of homologous recombination, RAD51, after irradiation, we can see that this is decreased in the BRCA1 deficient cells, but this is largely restored in the BRCA1 53BP1 deficient cells. BRCA1 deficient cells are hypersensitive to PARP inhibitor, which leads to a lot of chromosomal aberrations in a dose-dependent manner. But if you inactivate 53BP1 in gray, you do not get sensitivity to PARP inhibitor. And the double mutant mice, as I mentioned, do not get tumors. And very similar observation was made by Jas Jonkers, who studied the BRCA1 null allele, and which is, in the ES cells absolutely don't grow, but when you knock out or knock down 53BP1, amazingly they grow. And he also saw the restoration of this RAD51 foci, and homologous recombination was largely restored. And moreover, he found that in triple-negative breast cancers that there was a reduced survival when 53BP1 was deficient. So what we proposed is that really what BRCA1 does is it facilitates 53BP1 displacement. And this allows for resection to occur. So you basically, BRCA1 gets 53BP1 out of the way, so you can generate these these overhangs that can load RAD51 and allow for homologous recombination. Is that clear so far? Okay. All right. So in the absence of BRCA1, basically in the absence of BRCA1, this bad protein clogs the break site. You can't get the resection machinery to generate the single-stranded regions that are loaded by RAD51. And then in the absence of both BRCA1 and 53BP1, the resection machinery can go in there and generate single-strand DNA. You can load RAD51, and now homologous recombination is restored. It's a very complicated way, but to change homologous recombination deficiency to homologous recombination proficiency. So somehow, loss of BRCA1 is terrible, but the cells are in much better shape, and they're homologous recombination proficient when they lose 53BP1. And very simply, the major role of BRCA1 is to move 53BP1 out of the way. All right. So the other implication of this study was really that end resection, this conversion of double-stranded DNA to single-stranded DNA, is really a key determinant in the choice between non-homologous end joining and homologous recombination. You resect the DNA, and this is promoted by BRCA1. You perform homologous recombination. You don't resect, and this triggers a non-homologous end joining. So they're basically fighting at the break site. And whoever wins determines which pathway is utilized. OK, now there's one problem. We talked about resection, 5' to 3' resection, the generation of single-stranded DNA. But at that time, there was no way to precisely measure what we're talking about, end resection. The length, how long are these single-stranded regions? What is the rate of end resection? And how is it really influenced by BRCA1? So we figured that we should develop an assay to precisely measure double-stranded breaks and end resection in cells. And we developed a method called NSeq to map double-strand breaks and end resection. Basically, what we do is we place the cells in an agarose plug because we don't want to make the breaks ourselves by pipetting up and down. We remove protein and RNA. And if there is a break somewhere in the cell, we blunt that end by nucleases, and then we ligate an adapter, a biotinylated adapter, that we can then pull down and sequence. If we sequence that end, we get a left-ended read and a right-ended read as shown here, because a double-strand break has a left and a right end. And so if we use a restriction enzyme, and where we know that there's about 150 breaks that are generated by this restriction enzyme in the cell, if every cell has a double-strand break, as shown here, this has a 2 base pair overhang, so we would be blunting that end. We'd be ligating our adapter, and we would get basically 2 towers with a little gap in the middle reflecting those 2 base pair overhang that we had blunted. But if in the cell there is end resection, the generation of 5' to 3' and a 3' overhang, we would get it's very different. If we blunt the DNA here, different cells would have a different position here, and the first nucleotide that we sequence is quite different depending on the level of end resection. So basically, if there's no resection, you get a very sharp peaks at these break sites, and if there is resection, you get a broad signal. So for the first time, we could see really what BRCA1 and 53BP1, what the consequence of loss of BRCA1 and 53BP1 are. So this is one site, one restriction enzyme site. This is the wild type cell. You can see that the signal is much sharper in the BRCA1 deficient cells and much broader in the 53BP1 deficient cells. And this is another way to plot it. These are all 150 breaks. This is the length of resection, and each of the here is a different double-strand break. And you can see again that there's the loss of BRCA1 leads to very, very low level of resection, and the loss of 53BP1 leads to very increased resection, and the combined efficiency basically goes back to around wild type. And homologous recombination is restored. Okay, so in summary, 53BP1 counteracts BRCA1's function in resection, but there's another role for BRCA1 that was discussed in this meeting, and namely BRCA1 interacts with PALB2 and BRCA2 for the second step, which is loading of RAD51 onto the single-stranded DNA break. So the question that we raised is 53BP1 a block not only to end resection, but maybe it's also a block to the loading of RAD51 onto the single-stranded ends. So does 53BP1 impact the subsequent BRCA1-dependent loading post-resection? To determine this, we made a separation of function mutant of 53BP1 that we can separate these two functions here. And what one needs to know is that 53BP1 phosphorylation by the ATM kinase at the N-terminus at about 28 sites, these phosphorylations are necessary for its role of 53BP1 in blocking end resection. And so we mutated a single amino acid, serine 25, in 53BP1 that interacts with this protein p-tip, but leaves this interaction with this other complex, RIF1 and shieldin, intact. The loss of this this change of the single amino acid, serine to alanine in 53BP1, led to, there was no phenotype in the mice. So they were competent for class switch recombination, they were not radiation sensitive, and 53BP1 itself was recruited to the break sites. So then we performed in these normal mice this ultimate synthetic viability test, to ask whether this single mutation in 51 could rescue the lethality of the BRCA1 delta 11 allele. And what we found is, surprisingly, that that the single point mutation did lead to rescued the lethality, go from dead to alive with this single 53BP1 mutant. But they were not well. These double mutant mice died. They didn't die of cancer, but they died of what looked like many features associated with premature aging. They died pretty rapidly with aging features. And then we asked whether, like loss of 5301, whether homologous recombination was rescued in these double mutant mice. And to do this, we treated these mice with a PARP inhibitor in vivo. And the loss of 53BP1 combined with BRCA1, there is no impact of the PARP inhibitor because homologous recombination is largely rescued. But in these double mutant mice, basically these mice died within 9 days of PARP inhibitor treatment. And you can see that in the intestines were basically shot. The villi was quite shortened. So it had a really major impact in these double mutant mice. Also, we could test for homologous recombination deficient-like chromosomal aberrations. And BRCA1 deficient cells are high level of chromosomal aberrations after PARP inhibitor. And the double mutant mice also had quite a high level of of chromosomal aberrations. So think that homologous recombination, unlike the loss of 53-1, is not rescued. And moreover, we used our method NSeq to measure resection. And we could see that the resection levels, surprisingly, in this BRCA1 53-1 serine to alanine mutant was very similar to the BRCA1 53BP1. So levels of end resection were no different. However, the loading of RAD51 was highly defective in these double mutant mice compared to the BRCA1 53BP1. So how can the mice be alive if homologous recombination is really defective? So loss of BRCA1 and complete loss of 53-1 allows for the loading of RAD51, whereas in this mutant of 53BP1, you get high levels of resection, but the presence of 53BP1 still is blocking the loading of RAD51, and so you have a defect in RAD51, homologous recombination, and the mice are alive, but they're sensitive to PARP inhibitors. Okay, so that begs the question of how is RAD51 loaded onto resected single-stranded DNA in the absence of BRCA1 and 53BP1. I mentioned that BRCA1 promotes resection and 53-1 blocks this step, but also BRCA1 is necessary through its interaction of PALB2 and BRCA2 to load RAD51. How does this happen when you don't have BRCA1? And what we found is that there's an alternative pathway of RAD51 loading mediated by this ubiquitin ligase, this other protein, RNF168, that interacts with PALB2 and facilitates the loading of RAD51 in the absence of BRCA1. And moreover, the BRCA1 knockout cells are highly, grow very, very poor. The BRCA1 53BP1 double knockout cells, as shown by Jankers, there's no growth defect, but when you combine this with RNF168, you get a major growth defect, and that's associated with the loss of RAD51. So in the absence of BRCA1 and 53BP1, the way that RAD51 is loaded onto single-strand DNA is through this ubiquitin ligase RNF168. All right, so the therapeutic implications are, of course, that BRCA1 deficient cells are highly sensitive to PARP inhibitor because they have a defect in homologous recombination. And there are various mechanisms that lead to PARP inhibitor resistance. Probably the most prominent mechanism in the clinic is BRCA reversions that somehow restore the reading frame of the original protein. There is loss of PARP1 and PARG, shown by Jankers, and what we discovered is loss of 53BP1 can also restore homologous recombination and lead to resistance to PARP inhibitor. And there's a whole complex that associates with 53BP1 that gives a similar phenotype. And there have been many targeted treatments that you've heard about today that can potentiate, number one, PARP inhibitor sensitivity, but also be effective when you lose 53BP1 or shielding components. So Rogers' group and Simon Bolton showed that even if you lose 53BP1 and the cells are homologous recombination proficient, that if you inactivate this other gene, this chromatin remodeler, ALK1, you get sensitivity and you get cell death. So this is a way to generate toxicity or therapeutic treatment when you have a 53BP1 deficiency. And similarly, Yas talked about that ligase 3 deficiency leads to single-stranded gaps, which can be toxic in the BRCA1 53-1 cells. And Li Zhao showed that the combination of PARP inhibitor and ATR inhibitor leads to a defect in the RAD51 accumulation at these sites in the BRCA1 53-1 cell. So that ATR combined with PARP inhibitor would be effective. Just very similar mechanism as loss of RNF168. And we've also heard about Pol theta inhibitors, which are active not only in BRCA deficient cells, but in BRCA 53BP1 deficient cells by Neil today. And that was originally work from Chris Lord and Alan Ashford and Agnel Sphere. I think that one of the main issues is that, as far as I know, there's no targeted treatments for these reversion mutations. You can tell me if I'm wrong about that.
You have to come tomorrow to hear Stephen's talk.
Okay, okay. But this is there's a lot of work here, but less here when you get these reversion mutations. So this is a really important problem. And I want to talk about some unpublished work saying that we may be able to tune homologous recombination proficient cells to tune down homologous recombination. And so the starting point here is just a paper that I found where they mentioned that homologous recombination deficiency is inversely correlated with microsatellite instability and identifies immunologically cold tumors in most cell types. So, the claim is that homologous recombination deficient tumors are immunologically cold. I'm not sure if Serena completely agrees with that. I don't know if that's clear, but certainly microsatellite unstable tumors are generally hot tumors, and so they respond well to anti-PD-1 antibody treatment. So this original paper showed that mismatch repair deficient cell tumors were highly responsive to anti-PD-1 treatment. And this is largely because of the increased number of mutations associated neoantigens that are expressed in the absence of the mismatch repair. But even in the mismatch repair deficient patients, some respond, some don't respond. And so there were follow-up papers saying that the responding patients really had this feature of microsatellite instability. So if you divide the mismatch repair deficient cancers into those that are microsatellite unstable high and intermediate or low, the responding tumors are mainly microsatellite high. And again, this paper claims that homologous recombination deficiency is pretty much mutually exclusive with microsatellite instability. And if you remember Serena's talk, she gave an example, and she said that that's not really true, that you can have BRCA1 deficiency and mismatch repair deficiency at the same time. But this does suggest some kind of synthetic lethal relationship. And in that light, papers from 2019, a few years back, divided tumors into microsatellite stable and microsatellite unstable tumors and found that there was a really strong synthetic lethal relationship between the microsatellite unstable tumors and this single gene that stood out, and that's this Werner helicase. So the loss of the Werner helicase is incompatible with viability if you're microsatellite unstable. And we and other groups have shown that loss of Werner leads to the shattering of the chromosomes if in microsatellite unstable cells. So we take this microsatellite unstable cell line, KM12, and then with SH Werner, we knock down Werner, and we can see that the chromosomes are basically shattered into bits. If you take a microsatellite stable cell, the chromosomes are largely intact, even if you inactivate the Werner helicase. So, so similar to the BRCA-PARP story, the loss of Werner in microsatellite unstable cells leads to chromosomal breaks and cell death. But just to review, what is Werner? I said it's a helicase, so it unwinds the DNA, and that's and unwinding of DNA also, occurs during replication, transcription, recombination. This is a very basic process, and it's part of a family of RECU helicases that do the same thing. They are not essential for replication or transcription. What these helicases do is they recognize structures in DNA, supposedly, that occur during replication or transcription. So There are different structures of DNA, non-B DNA forms, such as DNA hairpins, cruciforms, G quadruplex, these funny structures that can occur. This is where these helicases get recruited and unwind the DNA so that replication or transcription can continue to occur normally. So that raised the question for us, where in the genome does the WRN act? What is the structure that it recognizes in microsatellite unstable cancers? And to address this, I showed you that the chromosomes get shattered in microsatellite unstable cells losing WRN. And so we performed our same assay, N-seq, to determine where are the breaks in the MSI cells lacking the WRN helicase. This is just a snapshot of a 2 megabase region, and these are our MSI cells. And in the absence of, in the presence of WRN, there's no DNA breakage. But when you knock down WRN, we can see these peaks here. There's the right end and the left end, and these are double-stranded breaks. And if you zoom in at one of these double-strand break sites, you can ask, is there a microsatellite around there? What is the motif? And we found, and basically in 90% or more, basically all of the breaks that there were TA dinucleotide repeats, which is a microsatellite. And if we zoom in here at this break, we see that it's not a clean break. It's not just those 2 towers. These look like highly resected DNA breaks that occur. So there's a lot of end processing. And so we asked whether these are actually, canonical resected breaks bound by RPA. And so we performed RPA chip to look at the localization of RPA, and we found peaks of RPA localization, and these localized precisely where the DNA breaks were located as well, as shown here as well. This is the zoom-in picture. So these breaks are highly resected. And we could also see resection as measured by RPA foci after irradiation. So you can see after irradiation that the MSI cells induce RPA foci, but this is much, much higher when you knock down WRN. So there's hyperresection measured by N-seq, RPA chip, and irradiation-induced RPA foci. But strikingly, in the MSI cells after irradiation, when you inactivate WRN helicase, we do not see any Rad51 foci formation. There's a lot of RPA at these breaks. It's highly resected, but there's absolutely no Rad51 foci. So we think that there is a defect in Rad51 filament formation in the microsatellite unstable cells lacking the WRN helicase. And so our model for what is going on is that you inactivate mismatch repair. It takes a long time, we don't know how long, to get an expansion of these TA repeats. They expand long enough so that there's a formation of this cruciform secondary structure that is recognized by the WRN helicase and is unwound by the WRN helicase. And if you lack the WRN helicase, this results in hyperresection, but defective Rad51 loading, defective homologous recombination, and genome instability. And we actually used PacBio sequencing to address whether there really was expansion of TA repeats that would be consistent with the formation of these cruciform structures. So we have a lot of sites that don't break, and we didn't see any length change in the microsatellite unstable cells, but in the microsatellite stable cells, but in the broken TA sites where there were breaks, we could see the expansion of these TA repeats. So the conundrum, what we don't understand very well, is this time dependence. So when you lack mismatch repair, at some point they become microsatellite unstable. And as I mentioned, the ones that are more microsatellite unstable are more responsive for anti-PD-1 treatment, and the ones that are less responsive. So how long does it take to get this, if you inactivate mismatch repair by itself, it does not lead to WRN sensitivity. It takes time. And this WRN dependence cannot be achieved in tissue culture. So if we generate MLH1 knockouts and grow them for 1 month, 7 months, or more than a year, and then we inactivate the WRN helicase, we'd see no effect on viability. That's very different from the microsatellite unstable cells where you inactivate WRN and you lose viability. So we cannot really reproduce in culture this change in the cell state between loss of mismatch repair and microsatellite instability for some reason. But in vivo, it happens. Oh, so the question we ask is, can we figure out a way to convert tumor cells to a microsatellite unstable-like state to generate WRN dependence and defective homologous recombination, Rad51 loading? Can we get around this somehow and produce WRN dependence and homologous recombination deficiency? And so we were intrigued by the literature about other triplet repeat expansions, like in Huntington's, where a group used replication stress, repeated pulses of acetylcholine, to generate expansion at these trinucleotide microsatellite repeats. They managed to get expansion of these repeats just by pulsing the cells with replication stress. So we performed the same assay. So we took our knockout cells, the MLH1 knockout cells that were not sensitive to loss of WRN, and we pulsed it with acetylcholine, let the cells recover for a few days, tested whether they were sensitive to loss of WRN, and repeated this experiment again and again. And what we found is, after about passage 14 or so, that the acetylcholine pulse cells suddenly became sensitive to the loss of the WRN helicase. And they were in a permanent state of sensitivity to loss of WRN because we don't have to pulse them anymore. They're in a state where you could now grow them perfectly fine, and then you inactivate WRN, and they're very sensitive. So it's a permanent, something changed in the cells. And these acetylcholine pulse cells, when you inactivate WRN helicase, they trigger a DNA damage response. There's chromosomal aberrations that are produced. And we saw hyperresection. So after irradiation, there is no difference whether you inactivate WRN or not if you don't treat with acetylcholine. But after this pulsing protocol, if you irradiate cells and in the absence of the WRN, you get this hyperresection, and you lose the ability to form Rad51 foci, very similar to the microsatellite unstable cells. So the acetylcholine pulsing does something to generate hyperresection in the absence of WRN, but complete absence of Rad51 foci. So so we used acetylcholine, but does generally replication stress induce Werner dependency? And we used 2 other agents, hydroxyurea as well as 5-FDU, which is used sometimes in the clinic for colon cancer. And we did this pulsing experiment, and we managed to get a completely different cell line sensitive to loss of WRN, and to get this in this permanent state of sensitivity. And interestingly, mild replication stress induces WRN dependency even in wild-type cells. So if you pulse wild-type cells, you also can get them to be sensitive to WRN. Not so much as the MLH1 deficient cells, but they definitely become sensitive to loss of WRN. And even in wild-type cells, you see a major reduction in Rad51 foci formation after this acetylcholine, repeated pulses of acetylcholine. We also tested whether we could induce this state of WRN dependency in a xenograft model. So in this case, we used this colon cancer cell line lacking MLH1, which was not sensitive to loss of WRN. We implanted it in the mice, and we treated the mice with 5-FDU, this replication stress-inducing agent, every other day for 2 weeks. And then we reimplanted these, the tumor cells back into another mouse. And meanwhile, we test for sensitivity to loss of Wern. And you can see that in the vehicle-treated cells, there's no effect on cycle 1, 2, or 3, these 2-week cycles. But in the tumor cells treated with 5-FDU, we can see that already by cycle 2 and certainly by cycle 3 that they become Werner dependent. So you can achieve the Werner dependence in vivo as well. And the loss of Wern in these pulsed cells leads to an HRD, homologous recombination deficient phenotype. In that, we can see radial chromosomes and chromatid breaks that increase with time. So this led us to test the idea whether these cells are actually sensitive to PARP inhibitor. These cells that were originally homologous recombination proficient went through this crazy pulsing, and they became sensitive to loss of Wern. They had a defect in RAD51 foci formation. Now, let's challenge them with a PARP inhibitor. And we can see that in the acetylcholine pulse cells, you get a major sensitization to PARP inhibitor upon Wern loss and the synthetic lethality here, which you don't see in the non-pulsed cells. So, and moreover, you lose viability when you combine the treatment with Wern deficiency and the PARP inhibitor deficiency. Okay, so how is the cell state changed upon these pulses of replication stress? So we're starting with a cell that is Werner independent. It's not sensitive to loss of Wern. It's homologous recombination proficient. We've done replication stress cycles, I guess that one actually does in the clinic. And the cells have become sensitive to loss of Wern, but they are homologous recombination proficient. Then we've knocked down Wern helicase. They exhibit hyperresection, but they become homologous recombination deficient and very sensitive to PARP inhibitor treatment. And so let's examine each cell state. First of all, did we achieve microsatellite instability? This cannot be achieved just by growing the cells in culture. So we did whole exome sequencing and found that that first of all, that there's that these cells have a signature characteristic of defective mismatch repair. But the acetylcholine pulse cells have many more point mutations than the non-pulse cells, which is maybe not completely surprising. They have a lot more indels, and by this MSI score, they have a high level of microsatellite instability from the acetylcholine pulses. We also performed RNA-seq analysis comparing the state 2 and the state 1, homologous Wern-dependent versus Wern-independent. There were a lot of changes, but the changes that we saw were very reminiscent of the changes relating to what I showed you before in the melanoma cells, the non-responding versus the responding cells. So the acetylcholine pulse cells had a lower level of expression of genes associated with wound healing, angiogenesis, and EMT transition, indicating that they might be more responsive, perhaps, we didn't test this, to immunotherapy. So they have more, they're highly mutagenic because they have MSI, express neoantigens, and have the signature associated with the PD-1, anti-PD-1 response. What about this state 3 when we inactivate Wern? So we performed RNA-seq experiments to look at the transcriptome 24 hours after Wern was depleted. And we found that, again, a lot of pathways were affected, but the major pathway, the most dramatic pathway that was affected were these so-called DREAM target genes that were downregulated in 24 hours after Wern depletion. So, and this just shows you 52 genes that are already quite downregulated 24 hours after the DOX-induced Wern downregulation here. So DREAM is a RB-like complex. It's a transcriptional repressor complex that represses cell cycle genes and many DNA repair genes. And we looked at cell cycle, and we could not see any defect in cell cycle progression at the early time points when you inactivate WRN. So at 24 hours and 48 hours, eventually the cells will die 96 hours later. But within 24 hours, there were plenty of cells and very similar percentage of cells in S phase. However, when we looked at homologous recombination associated genes, like RAD51, BRCA1, BRCA2, et cetera, they were highly downregulated within 24 hours. Genes involved in holiday junction resolvase genes were also highly downregulated already by 24 hours. And the Fanconi anemia pathway was also severely impaired upon WRN loss in these acetylcholine pulse cells only 24 hours after loss of WRN. Moreover, we could see that the RAD51 protein levels were also decayed very, very rapidly upon WRN deficiency in the acetylcholine pulse cells, which makes sense because we didn't see RAD51 foci. It's because the protein levels are quite low. Okay, so we have these homologous recombination proficient cells. We have replication stress cycles that induce high levels of microsatellite instability and perhaps expression of new epitopes. That's a good thing. And when we inactivate WRN, we convert these cells to homologous recombination deficiency and make them sensitive to a PARP inhibitor, and maybe they would be responsive also to anti-PD-1 treatment because, after all, they're MSI-like. So we have three independent ways, perhaps, to have a treatment for homologous recombination proficient cells. Very complicated, but this is one way, at least, that we have managed to convert homologous recombination proficiency to deficiency, and from microsatellite stability to microsatellite instability. Okay, so that about does it, and I just want to acknowledge with a photograph of the people in my laboratory throughout the 12 years that have been there that have really contributed to this work. I'm not going to name names except for Elsa Callien and Nancy Wang, who have been with me from the very beginning and have withstood me through time. So I thank them and thank you for listening. And thank you very much for this award.
Okay, so we certainly have time for questions. So we've got Kate, so Jess will give the first microphone to Kate Nathanson.
Thanks, Andre. So we actually had several publications showing that BRCA1 and 2 mutant tumors look to be immunologically cold as well, both breast and ovarian cancer with slightly different mechanisms. So my question actually is, based on your work, you've gone through all these. How do you get those immunologically cold tumors to become potentially more immunologically responsive? That to me is the challenge.
Yeah. So if what I showed can be achieved in the clinic, if you could convert microsatellite stable cells to microsatellite unstable cells, you might have a chance to make change from to immunologically cold to immunologically hot tumors. So that's what we have achieved in this strange, in this tissue culture with repeated replication stress. And
can I ask a question about that? Sorry to interrupt. Was there something, you mentioned that you used both 5-FU and I think, I forget.
And hydroxyurea, and acetylcholine.
Is it anything that can do that? Because, in clinic, I guess anecdotally, we don't necessarily see that. We give lots of chemo to lots of metastatic patients, and they don't necessarily become then immunologically sensitive. So do you think it's a specific thing you have to give, or do you have to then deplete Werner? Like, how do you? Yeah,
I think that we just have to collaborate and test the different compounds and the protocols I mean, how many pulses do you have to give? How do you do it?
Yeah.
But at least in tissue culture, we achieve it. We don't know which compounds actually do it. But the general theme is repeated replication stress-inducing agents.
And it may be a schedule effect, right? I mean, because we all know we don't really know how to schedule chemotherapy. So there may be something special about how to do that.
Yep, we use very low doses, and then when the cells recover after a couple of days, we do it again and again and again.
Gotcha. Okay. Yeah. All right, Kara.
I was just curious if you could comment on the mechanism of how Werner affects RAD51. I guess you looked at maybe it's not loading, right? Necessarily, it's the protein levels, and it's a helicase, so I'm having trouble wrapping my head around it.
So the protein levels go down, and I think there's a major impact on E2F target genes, including RAD51. So the expression goes way down, and that's why we lose RAD51 foci, as well as, I'm sure, many homologous recombination proteins.
So, Andre, very interesting, and I'm just trying to connect those two and building on this. So the dream complex loss of it, my understanding, will allow expression of these genes, RAD51 and others, and even to load even in G0 cells, right, the Barry Schleckman paper. So this loss of HR in the absence of Werner's, and you need microsatellite instability as well. So are you having expansion of repetitive elements around this in a way that is silencing or enhancing the dream complex and working? And basically, you're dampening down all these HR genes because you have repetitive DNA going, expanding, you're getting heterochromatization, and you're turning down HR overall. And I don't know if that's something that you've looked at, but...
Yeah, I mean, we have done exome sequencing, and we've also done PacBio sequencing. One thing I can say is that this mechanism is different from the TA expansion. We know that that does not happen. So there's different ways to get Wern sensitivity through microsatellite instability. This is a different way, and it could be related to what you say, but we don't know.
Do you have any repetitive element expansion around sort of E2F1 targets?
Yes, we should look at that. We haven't looked at. We definitely have microsatellite instability, so we get expansion in a lot of indels, but where they actually occur, we should look at that. Because you're right, we don't know really how this is affecting E2F target genes, but it is. We just don't know how.
Go ahead.
So I'm not sure if I missed it, but can you say how you can imagine getting a therapeutic window here? If you're taking a essentially wild-type cell in the sense that it's HR proficient, how do you get specificity for the tumor cells if you think about this in a clinical setting?
It's true that in theory, all dividing cells would be impacted.
Like we do with chemotherapy.
Yeah.
That's
like chemotherapy. But if you do theoretically, if you do chemotherapy correctly, you can convert to microsatellite unstable and Wern sensitivity.
Or maybe there's another alteration that makes them more sensitive, like the chemo. Yeah. Chemo does.
We need to do more experiments in vivo, I think.
So we're going to go Ronnie, and then Andrew, and then Lee. We got Lee, don't worry. Ronnie first.
So along the same lines, you talk about pulsing with acetylcholine to cause the replication stress. But what about in cancer cells that have replication stress already? Does that create any dependency? I'm thinking about cyclin E overexpressing or amplified cells where you have this sort of chronic replication stress. Is that different, or is that potentially leading to similar?
That's a really good question. And all I can say is that, in any cell, BRCA deficiency is associated with homologous recombination deficiency, any cell type, right? But Werner deficiency is a kind of special. Its loss doesn't affect all cell types. So if you take stem cells, you lose Wern, they're perfectly fine. But if you differentiate them, they become sensitive to Wern. So there could be certain cell types, maybe some cancer cells that have a lot of replication stress. Maybe they are sensitive to loss of Wern. We just don't know. But I do know that it's not like BRCA. There are certain cell types, particularly those that are telomerase positive cells that are much less sensitive to loss of WRN than telomerase deficient cells. In fact, Ron DePino a long time ago combined telomerase deficiency and WRN deficiency, and he found basically that generated WRN-like aging syndrome. But yeah.
Andrew.
Andre, apologies if I've got this the wrong way around, but as I understand it, part of what you may be suggesting one might engender is an MSI high phenotype. How long do you think you would need to do that for to generate the level of tumor mutational burden to create checkpoint inhibition sensitivity? Because truly MSI tumors have been MSI for a long, long time to develop that level of neoantigen burden. Yeah. I just wonder how long you've got to do it for to get to a targetable level of neoantigens.
Right. So, we know that we get how long it takes to get WRN sensitivity, and we would have to do the actual experiment in vivo, which is definitely doable, to see how long it would take to get, or if they become responsive to anti-PD-1 therapy. It's an experiment that we just haven't done yet.
All right. Lee's been patiently waiting, but he's finally got the microphone.
Yeah, great talk, Andre. So I wonder if loss of WRN is the only way to shut down HR genes in MSI tumor, right? So if you increase the instability of microsatellite repeats in different ways, do you still get the same outcome? Is it something special about WRN, or is it something about the instability in those repeats?
I can tell you that another experiment they're doing is, now that we have acetylcholine pulse cells that are sensitive to WRN and the unpulsed cells, then we do a CRISPR screen to see what besides WRN leads to loss of homologous recombination and toxicity. WRN is definitely essential in that state, but there could be other genes that do the same thing. We don't know.
Yeah, the questions keep on coming. Go ahead.
That's a very nice talk. Quick question. Based on your signature that's associated with this, the EMT signature, I'm wondering whether you see a difference with metastatic versus localized response. If you were picking cells that have that capability of migration versus not, how that might respond differently for your MSI.
Can you repeat the question?
So part of your signature was a change in EMT.
Yeah.
And so that's associated with a metastatic spread signature. So I'm wondering whether or not cells that have more capability to spread versus not would respond differently.
Yeah, I mean, we have that signature, but we actually have to test whether there really is EMT. This is just an RNA-seq pathway. So I can't say that there is an impact on wound healing or we just have to, this is a hint, and we have to take a look at if it's actually true.
Bob.
Thanks, Andre. Congratulations. Okay, I have just a crazy question, and you can just tell me it's nuts. But when I saw the cells being pulsed and everything's fine, and then the next cycle they're not fine, that feels like you've screwed up the telomeres. And I wonder if you've actually looked as you went from left to right, if that biology is...
Do we still have the slides?
Okay.
Well, the answer is yes. We screwed up the telomeres.
Oh.
So as a function of pulse, the telomeres go like this. I don't have to show the slide. We see a loss of telomeric DNA. Now, how that contributes to the WRN sensitivity, I don't know. But there definitely is a loss of telomeres at each pulse. And there is sort of what you mentioned, a kind of a sudden threshold. Passage 14, boom. Yeah. They suddenly become, that seemed almost like something happened in that cell division.
Okay.
But there's a gradual loss of telomeres.
So is that just true unrelated, or does that have something to do with the emerging phenotype?
We see telomere fragility, so there's a problem. So WRN, it recognizes DNA structures. There's funny structures, G quadruplex structures at telomeres. So if you so maybe something happens with those structures that is altered by the replication stress that now really requires WRN. When you don't, when you lose the when you lose telomeres. But we don't know how that phenotype, which happens, how it really contributes to the Wern dependence.